Welcome everybody. So summer has just begun, even though it's not that obvious in Britain because with the weather, but I assume that we will all be putting on sunscreen this summer, maybe once you leave Britannia. So I'm going to be talking about the effects that sunscreen have on marine bacteria. So this is a popular image that we may be familiar with. So it quotes, just because you can't see it doesn't mean it isn't there. But when we think of marine pollution, the first thing that jumps to our minds is plastics. Well, there's a group of pollutants called emerging pollutants, and the EU's advisory board, the JCR, reported last year that there are 2,700 potential marine contaminants that are largely unregulated and the potential effects are not well understood. So for these reasons, it's a growing concern for the ocean. So although we have wastewater treatment plants in place, these emerging contaminants are not being fully removed. They get this name because they've only been identified in the last 15 years, thanks to improvements with analytical chemistry, namely liquid chromatography coupled with mass spectrometry, that's able to detect the emerging contaminants in the nanograms per litre range. So a group that falls under this category of emerging contaminants is called pharmaceutical and personal care products. So some examples of these are plastic microbeads, fragrances, preservatives, insect repellents, and the one that I'll be talking about for this study is UV filters. So a study has been performed on different marine organisms, from algae, fish, phytoplankton, and coral reef that's shown negative impacts due to UV filters, from growth inhibition, endocrine disrupting effects, coral bleaching, but there hasn't been much done in literature on the effects that it could have on marine bacteria. So marine bacteria are one of the most abundant organisms in the ocean, and if you take 1 ml of coastal seawater, you can find up to 1 million cells of bacteria. And they're important for several reasons. So they are a crucial player in the marine food web, with mostly protozoa feeding on them. They decompose organic matter to nutrient minerals. They're involved with different symbiotic relationships, and they're also involved in different biogeochemical cycles. So it's important for us to understand how are emerging contaminants affecting marine bacteria. Also, sunlight radiation. This is an environmental variable that influences the biology in the ocean, but also impacts emerging contaminants. So this is a UV filter that's undergoing a cis-trans photoisomerization due to UV radiation exposure. And although a lot of effort has been made on the occurrence of UV filters in the ocean, there's not a lot understood about the transformation products of UV filters due to solar radiation. But I won't be talking about the chemical transformation of these UV filters, but I'm interested in how the toxicity is impacted by the alteration of these UV filters due to photolysis. So all of this brings us to want to investigate the impact on organic UV filters on heterotrophic marine bacteria combined with solar radiation. So there's 2 phases of the study. The first phase looks at the screening. So we want to see which bacteria are sensitive to the UV filter. Once we find those, we then expose them to the combination study that examines the toxicity of the UV filter combined with solar radiation. Just to explain the combination study a little further, there were 2 questions that were asked. So first of all, we wanted to know, how is the UV filter's toxicity altered when previously exposed to solar radiation? And also, how was the UV filter's toxicity altered when the bacteria were also present to the solar radiation? This was examined on the bacteria at their 2 phases, their growing log phase and their stationary phase. So now I will talk about the experimental approach. So the screening phase. So a strain panel was obtained by isolating 25 marine bacteria from the northwest of the Mediterranean Sea, and they were exposed to a concentration of 1,000 micrograms per litre. And a microplate reader was used to measure the optical density and to obtain graphs like this. So when we saw that to find if the bacterial growth efficiency was affected due to the presence of the UV filter. These are the 5 UV filters that were chosen for the study based on occurrences that are found in the ocean. I'll be using the abbreviations in this study. So when the bacteria did show sensitivity to the 1,000 micrograms per litre, the bacteria was then underwent a dose response analysis from 100 micrograms per litre to 4,000 micrograms per litre. And then after the screening was performed, that strain was then used for the combination study. So for the first question of how the UV filter, when it was previously exposed, we had 3 treatments. So the treatment that contained a UV filter and was exposed to an artificial solar lamp for 5 hours. Then the UV filter that was not exposed and a control that did not contain a UV filter. And the medium was artificial seawater. So first of all, exponential. When the bacteria were in their growing phase, the cells were inoculated into the medium that was previously exposed, or the 3 treatments that I just spoke about. And this medium also contained a carbon source so that the bacteria could continue to grow. And then this growth was monitored. And the different treatments were compared from the growth rate. Then for the stationary phase, the cells were isolated, removed of any carbon source, and then they were inoculated into the treatments that did not contain a carbon source. Then the percentage viability was measured by plating the surviving coliforming units, and we compared the survival rate from T0 to over 24 hours, and we compared that between the different treatments again. So the second question was the direct effect. So what was the effect when the bacteria were also exposed to the solar radiation? And they look like potatoes. Actually, that's bacteria in the flask. So same idea again, except the growth was just measured over 7-hour period, and the percentage viability was measured over a 5-hour period. So the statistical approach that was carried out, when we had a full growth curve, a generalized additive model was used to compare the different treatments. And then when the 7-hour growth experiment was performed, just a generalized linear model was performed, and the data was transformed depending on the residuals distribution. And then for the percentage viability, the Tukey comparison test was performed. So now for the results and discussion. So first of all, the screening phase, when the bacteria are exposed to the UV filter. So this is not a very legible slide, but it's just to show you the strain panel. And the bacteria that were, the orange box is the bacteria that were susceptible to the UV filter. And 23% of these bacteria showed sensitivity to one or more UV filters. And just to note that none of the gamma proteobacteria showed any sensitivity to any UV filters. And these bacteria can also be found in polluted waters. So I don't know exactly why, but they're more resistant to contaminants. The UV filters that were used was mostly BB3 and EHMC that predominantly showed toxic effects. And just to note that the 4-MBC showed no toxicity, even though in literature it does show toxicity effects to different marine organisms. So the reasons why there's different sensitivity to different UV filters and different bacteria is because the UV filters are different chemical compounds, and the bacteria have a different physiochemical structure. And we can see this when we look at, this is a dose response curve, and we can see this is the same strain exposed to 2 different UV filters. And in the first one, the black line is a control containing no UV filter. The dark purple line is a higher concentration. And we can see in the first one a reduced bacterial growth efficiency. But then when the same strain is exposed to HS in the lower graph, there's a longer lag phase here. So the bacteria are taking time to adapt to this UV filter before it grows again. And then with other strains, we see that there was different sensitivity to the different UV filters, with some showing mortality at the higher concentration. So those strains were then underwent the combination study of the UV filter and the solar radiation. So first of all, when the UV filter was previously exposed to the solar radiation, we obtained graphs like this. So at the side here, this legend is the UV filter and whether it was exposed or not to solar radiation. So we see that the red line is where the bacteria growth efficiency was reduced the most. And this is when the UV filter was not exposed to solar radiation. So this would imply that the toxicity was actually reduced when the solar radiation was present, indicating that the parent molecule is more toxic, whatever alteration happened to the molecule. So this was observed for the UV filter HS and EHMC. And in literature, photolysis does occur when these UV filters are exposed to solar radiation, but we just didn't know the effects on the toxicity. So when the bacteria are exposed to the solar radiation also, we saw graphs like this. So the predominant reducer was due to the solar radiation, but actually more significance was seen when there was a combination of UV filter and solar radiation, when the bacteria were in their exponential growing phase. Again, this was seen with HS and EHMC, showing that more significance was observed in the combination study. So, but with the bacteria, Epibacterium immobile and BB3, different results were observed. So when the bacteria was previously exposed to solar radiation, we saw graphs like this. So no matter whether the UV filter was exposed to solar radiation or not, the UV filter reduced the growth of the bacteria. And again, in literature, this molecule is seen to be more stable, so the UV radiation didn't alter it. So when the bacteria were exposed to the solar radiation, we reduced the concentration of the UV filter to not mask out any combination effects. And the same thing was observed again. This is just a box plot, and it showed here that in the presence of the UV filter, there was a reduced growth rate, regardless of the solar radiation or not, for the exponential phase. But then for the stationary phase, a different story is observed. So here is the Epibacterium are exposed to the UV filter, but they were not affected by their presence when they were in stationary phase. So this indicates that when the bacteria are in stationary phase, they're more tolerant to the UV filter. But when combined with solar radiation, they were sensitive to the UV filter. So the UV radiation actually increased the sensitivity to the UV filter, and we saw it's again seen in literature that it can reduce the bacteria's metabolism, making them more susceptible to contaminants. So now to conclude. So for the screening study, it all depends. It depends on the dosage and the UV filter that was used, because there's so many different factors with the chemical compounds and for the strains as well with the physiochemical structure. And also the growth phase depends. But what is a bit more concrete from this study is from the combination study results. So it hasn't been done before with the combination of bacteria, solar radiation, and UV filter study. And this study concluded that the HS and EHMC toxicity was reduced once exposed to solar radiation. And this is a positive combination effect that is observed. we saw that the BB3 toxicity was not altered in the presence of the solar radiation, and that solar radiation can increase the bacteria's sensitivity to the UV filters also, and this is a negative combination effect. So the study is continuing looking at different strains and UV filter combinations. Along with this study, flow cytometry analysis was performed to look at if there was any morphological changes on the bacteria at a lower dose of UV filter. The PhD student Clement Lozano will be doing chemical analysis to see any transformations of the UV filter, and he'll also continue to look at the proteomic studies to see how these bacteria are, what mechanisms they're undergoing to be able to defend themselves against these contaminants. And I just want to acknowledge the LBBM lab for taking me on as an intern, and to Clement and Philip Le Baron, my supervisor.